So what we're going to look at now follows on from the carbohydrates lesson, where we're now going to look at the biochemical tests for two types of carbohydrate. The first we're going to look at is something known as Benedict's test, as it says up there. And the key here is that this is a test for both reducing and non-reducing sugars. Now, what do I mean by this? And it's quite important that you understand what we mean by a reducing sugar. And essentially, a reducing sugar quite literally means that the sugar has a functional group within the molecule that is able to reduce another compound. That's it, quite straightforward. And what we mean by reduction in this case is the chemical term. Don't forget we can use oil rig as an acronym. Oxidation is loss, reduction is gain. So we can split this into two essentially. Oxidation means loss of electrons, reduction means gain of electrons. Therefore, if you can reduce another compound, electrons will be given to this other compound. So the compound that we reduce gains electrons. So essentially, if we have our carbohydrates, let's say it's glucose in this case, if it is a reducing sugar, which glucose is, it can give electrons to another compound. Here we'll just call it compound A. And compound A gains the electrons and becomes compound B. So the electrons are sort of stuck on it like that. And therefore, in this diagram, we can see that glucose has lost electrons. Therefore, this has become oxidized. Oxidation rather, is loss of electrons. Meanwhile, our compound here, compound A, has been reduced. It's gained electrons. So, what makes a reducing sugar, or a sugar able to act as a reducing agent? The answer, quite simply, is you need a free or an available either aldehyde functional group or a ketone functional group. So in your chemical structure, you either need to be an aldehyde or a ketone. And of course, we've come across these last lesson. We know that both glucose and galactose are aldose sugars. They have free aldehyde groups within their chemical structure. Meanwhile, the ketone functional group here is present in fructose. These guys that we've come across before, these are all monosaccharides. Remember, glucose, galactose, and fructose are all reducing sugars. So, let's look at this here. This is kind of what's going on. You may look at the structure of glucose like this, and you may question me and ask, where is the free aldehyde group? Or if it was fructose, where is the ketone group? It doesn't appear to exist in the cyclic form. And of course, the answer is it doesn't. You're correct. Because when or where the aldehyde functional group used to exist was on carbon 1, which is over here. In the linear form, This was the carbon that had the double bond oxygen and the hydrogen. The double bond oxygen, in the process of becoming cyclic, became a hydroxyl group, OH. So we have lost the aldehyde group. So how does this make glucose reducing? Well, the answer is this isn't a reducing sugar. So cyclic glucose is not. But don't forget, we also have linear glucose, which does contain our free aldehyde group and is therefore reducing. So don't forget that these two compounds, these two forms of glucose, we have cyclic here and we have linear here, are in equilibrium with each other. And this is a poorly drawn equilibrium arrow, because actually, if you remember back to what we said last time, most of glucose is cyclic. It's around about 90 to 95%. Only about 5% is actually linear, but that's enough. That's enough to allow a solution of glucose to act as a reducing agent, to reduce some other compound. So it's not the cyclic glucose that's going to be doing it, it's the linear glucose. And this will be the same for fructose, where you have the ketone functional group in the linear form. So it is absolutely the linear version of these sugars that is giving the reducing or sort of has the reducing capacity. The cyclic form does not, despite it being the more common of the two. So if we were going to actually draw this equilibrium arrow, it shouldn't look like that. It should be massively skewed towards the cyclic form. So we should have a big arrow going this way and a very small arrow going this way. So let's imagine we've got our linear form. It's 2 to 5% of the total. And what's the point? What's this going to achieve? Well, we can give it a compound that can be reduced, and we give it a compound that contains copper 2 plus ions, and we have copper 2 plus ions. Sorry, my computer's being a bit weird. In the form of a solution of copper 2 sulfate, 2 in Roman numerals denoting the oxidation number, or essentially the charge on the copper ion. So copper 2, if I'll draw that arrow a bit better. Copper 2 sulfate solution. So this is aqueous. And if we react this with our reducing sugar, in this case linear glucose, the aldehyde group over here can give electrons to our copper 2 sulfate. And what happens is the copper 2 sulfate, the copper 2 plus ions, are reduced. They gain electrons. First of all, they become sort of copper 1 plus, and they then form with oxygen a solid precipitate, so this would be solid here, of copper 2 oxide. Rather, copper 1 oxide. So Cu2O means that copper has a 1 plus charge in this ionic solid. So we can see a color change. If we have a solution of copper 2 sulfate and we react this with our free aldehyde group, in other words, with our linear glucose, we produce a precipitate of copper oxide, copper 1 oxide, copper 1 plus ions here. So hopefully you can see the overall reaction is, in terms of ions, that copper 2 plus, which is blue, gets reduced into copper 1 plus, which forms as a solid ionic compound, as a red precipitate. And in the process, if the glucose is reducing something, it itself must become oxidized. Don't forget, we said up here. right? The glucose itself must become oxidized. Therefore, if we oxidize an aldehyde group, we become a carboxylic acid. So this functional group, C double bond O OH rather than just H, we've gained oxygen here. Okay, it's become oxidized. The other definition of oxidation, of course, being gaining oxygen. This is now an oxidized aldehyde, which is a carboxylic acid. Now, that's kind of less important for what we're bothered about. What we're bothered about is the color change. And the overall name for this is Benedict's test, because the copper 2 sulfate solution here we refer to in biology as Benedict's reagent. And you can write this in an exam. Benedict's reagent. It's a solution essentially of copper 2 plus ions, copper sulfate. So let's look at how this would work. This is how you would carry out the test. You have a liquid solution, an aqueous solution of your carbohydrate, or the solution that you want to test to see if it's made up of a reducing sugar, something like glucose, or indeed fructose or galactose. And we'll get back to that in a minute. We add double the volume. You can actually add the same volume. In this case, we're adding double. It doesn't really matter too much what you say. Either add an equal volume or add 2 times the volume of Benedict's reagent. Notice that this is a blue aqueous solution. What you then need to do is you need to heat it up in a water bath. You can put it under a Bunsen burner. Don't say that in the exam. Say that you put it in a water bath and we heat it to about 60 or 70 degrees Celsius. We have to heat for around about 5 minutes, which is what you would have done in the practical. And then we look for a color change. And what you'll notice here is it doesn't immediately go red. And the reason being is, although over here oops, that's not red. Over here, we absolutely would have our copper 1 plus solid, our copper oxide, Cu2O solid, produced. It's kind of a stepwise change. And it depends on how much glucose or how much reducing sugar, what the concentration of reducing sugar is in your initial sample. If you have none, then you remain blue. If you have a little bit, then you go green. Eventually, you then go sort of yellowy orange if you have a little bit more. And if you have loads of reducing sugar, you form a brick red precipitate rather than a yellow or a green precipitate. The key thing for an exam is to say that the color change occurs because we form a precipitate, a solid precipitate, and you have to name the color. So in an exam, if it's asking you what the positive result is, just say it's a brick red precipitate. Solid brick red is the color. It may accept red in a mark scheme, but brick red is a better term. But if you don't have enough reducing sugar in the solution, you will get a green precipitate or a yellowy orange precipitate. So that is how you carry out the test. So if you're going to write this in an exam, you need to say that you have a liquid form of your sample. You add Benedict's reagent, either an equal volume or double the volume of Benedict's reagent to your sample. You heat in a water bath for 3 to 5 minutes to about 70 degrees Celsius, 60 to 70 degrees Celsius. And you then observe, and a positive result is that you get a color change. And you have to say in an exam what the positive result is. So I'm going to put a little nota bene here, NB. Always state the positive result. And in this case, it is a brick red precipitate, PPT, brick red precipitate. You have to say what the positive is. There's always a mark for stating when you're asked to write the steps for this test, there's always a mark for stating what the positive result is. Very important. So that's how you test for a reducing sugar. Now, I'm going to give you some rules. Very easy to remember rules, which is, what is a reducing sugar? How do you know? Well, as we've seen, all monosaccharides that we've come across are reducing sugars. And that is because, as we've seen with our examples, we have a free, in the linear form, aldehyde or ketone group that are able to reduce compounds and become oxidized themselves. FYI, for those chemists amongst you that are going, well, hold on a minute, if we've got fructose and we've got a ketone group, surely that can't be oxidized because you cannot oxidize ketones any further. And you would be correct. And I'm not going to go into the answer. If you're interested, you can either email me if I teach you. If I don't teach you, I guess message me on YouTube or something. There is a reason that this works. If you wish to look it up, I will just give you a hint. It's called tauto isomerization. So it's something called tauto isomerization of ketones allow this to actually still act in a reducing capacity to reduce our copper 2 sulfate into copper 1 oxide, Cu2 plus into Cu plus. So don't worry about that. You will never get asked that, but be aware and become oxidized itself. But there is a sort of slight word of caution there, which is it's because of something called tauto isomerization, but that is far beyond the A-level requirements. But all monosaccharides are reducing. So the monosaccharides we've come across are glucose, galactose, and fructose. Because glucose and galactose have aldehyde groups, fructose has ketone group. Right, moving on. The other one is that all disaccharides, actually, I'm not going to say all, most disaccharides are reducing that we come across. And of course, the disaccharides that you have to know are lactose, maltose, and sucrose. However, there's an exception. Lactose and maltose are reducing, sucrose is not. Sucrose is not a reducing sugar. So that is the exception that you need to learn. Sucrose is different. And the reason why it's different, which will hopefully help you remember that this is not a reducing sugar, is if we go back to this from our previous lesson, if I kind of get rid of this stuff, is because lactose and maltose look very similar in terms of their structure. We have two hexagons, and these, of course, as I've just mentioned, are both reducing sugars. The difference is sucrose. And the reason, which is why I laboured the point last lesson, as you can see, is because of this. It's because sucrose is made up of glucose and fructose. Fructose is it has a different shape, right? It's a structural isomer, don't forget, of glucose. It's got a sort of pentagon style shape, not a hexagon. Now, I'm being quite vague in my sort of terms there. If I taught you in class, then I went into a lot of detail about acetyl groups, hemiacetyl groups, and that kind of thing. Don't worry about it. You don't need to know it. So for the sake of this video, I'm not going to cover it. all you need to remember is that because of the fructose group being weirdly shaped, essentially, it's sort of a pentagon, it messes up the structure of sucrose, meaning now, because of this, this is not a reducing sugar. And again, the keen-eyed amongst you will be looking at this and you'll be going, well, hold on a minute, Dr. Hayes, there are no aldehyde groups here. And with fructose, there's no ketone functional group. So how is this even a thing anyway? As I said, don't worry about that. They can still be converted into their linear form. Right. But fructose here kind of messes all this up. So I'm not going to go any further than that. If you're interested, email me or send me a message. But that's kind of the point that I want to get across. Of these three disaccharides, it is lactose and maltose that are reducing. So these guys will give a positive test, a positive Benedict's test. Where we will go from a blue solution to, or we will form a, that's not even red, is it? I've been drawing it in pink. Whoopsie. A brick red precipitate. And of course, if you're really good, we will say that this is a solid precipitate of copper 1 oxide. So this guy, however, will not do this. Sucrose, this will not give a positive test. This will give a negative result. Negative Benedict's test. In other words, in our tube here, where we've put our Benedict's reagent and our sample, it will remain, after heating, it will remain light blue. Light blue solution, no precipitate formed. So, the reason I point this out is we need to test now. How do you test for sucrose? Because this is really important, just from a general sciencey point of view. This is really important. We cannot use a negative result to infer anything. And that is generally true across biology. We cannot simply do the Benedict's test as described, and if there is no change, what we cannot do is say, oh, well, it must be sucrose, or it must be a non-reducing sugar that's in there. Because the alternative here is that the whole thing just didn't work for some unknown reason. Maybe the water bath was not warm enough. Maybe it was too hot. Maybe whoever made up the copper sulfate solution, the Benedict's reagent, didn't do it properly. Lots of reasons. Or maybe you've got contamination or whatever. You cannot simply say that because there was no color change, there must be a non-reducing sugar present. What we have to do is now do something else. So let's just do this as a separate thing. How do you test for a non-reducing sugar? So reducing sugars, as we've seen, fairly straightforward, standard Benedict's test, we get a positive result. If it's a non-reducing sugar, we carry out Benedict's test. Oops. That's the first thing we do. So you need to write in an exam how we do that. So add your Benedict's to your sample, heat it in a water bath to 60 or 70 degrees, look for a color change. And we have to say that there is no color change. Because it's a non-reducing sugar, so we think. But as I've said, we cannot simply use this. We cannot just say, oh, well, it must be a non-reducing sugar. What we have to do now is we have to move on to another step here. So this is step 1, carry out Benedict's. In an exam, I would maybe suggest writing that out. But step 2, we now need to, if we have something like sucrose, and if I just go back to it up here, if we have sucrose in our mixture, Essentially, the negative result has ruled out both of these guys, right? So it's not lactose and it's not maltose. What we now need to do is prove that it is sucrose or at least some other non-reducing sugar. So what we need to do is first hydrolyze the glycosidic bond here. And we do that using acid. So we can use acid, dilute acid. So acid hydrolysis. Hydrolysis, don't forget, is the posh word for breaking a bond, breaking apart these two molecules. And it's going to break here the glycosidic bond. Acid hydrolysis of the glycosidic bond. And hopefully what you appreciate is if we break this bond, we're going to liberate our two monosaccharides, whatever they are. Well, we don't technically know what they are, but if it's a disaccharide, if it's a non-reducing one, then we will split up and we will liberate our glucose and we will liberate our fructose. If it's sucrose in this case. And of course, both of these guys are themselves monosaccharides, and all monosaccharides are reducing. So this is what we need to do. We need to add dilute hydrochloric acid. You could write this as hydrochloric acid aqueous to the solution. And we need to heat it up in the water bath for a further 3 to 5 minutes. What we then need to do is we need to let it cool to sort of room temperature and add an alkali to neutralize this acid. So what we add is usually dilute sodium hydroxide, so sodium hydroxide aqueous, dilute sodium hydroxide to neutralize the acid. And we need to do that. We need to neutralize the acid because Benedict's does not work in acidic conditions. So once we've done that, essentially in our tube, in theory, if we did have a non-reducing sugar, right, if we had a disaccharide, and I'm just going to draw them as two blobs now just for ease. Oops, don't know what happened there. If we have a disaccharide like this, what we've done is we've acid hydrolyzed, we've broken up the glycosidic bond, and we have liberated your monosaccharides. This would be glucose, and if it's sucrose, of course, this would be fructose. Of course, we don't technically know that, but the idea is that we're liberating monosaccharides. Both of these are now reducing. Yeah, both of these are reducing sugars. So we then repeat Benedict's again. So we add an equal volume, or 2 times the volume of Benedict's reagent. We then heat it up again in a water bath. It's always worth putting the temperatures, so roughly around 60 to 70 degrees C for about 5 minutes. And we should now get a color change. We should now get a positive result for reducing sugars. Therefore, we should get a brick red precipitate, or potentially green or yellow, orange, depending on how much we've got, how much reducing sugar is present. So hopefully what you can see is this is now a positive result. This is something we can actually see. And what it proves is that if we have a brick red precipitate here, that means we must have monosaccharides liberated using acid hydrolysis. And if we have monosaccharides available that are reducing, that means they must have been connected in a glycosidic bond initially. So we must have had a disaccharide present, and that disaccharide must have been a non-reducing sugar. So if you were given an exam question and you had lactose, maltose, and sucrose, we can absolutely then say that you must have had sucrose in that solution. Of course, there are other disaccharides that are non-reducing as well, but these are the only three you need to know. So hopefully that makes sense. It's a little bit more complicated, not too much. Essentially, we need to break the glycosidic bond. And actually, I would say that in the exam. So if you're writing this as an exam answer, absolutely say that you add hydrochloric acid to break or hydrolyze the glycosidic bonds and liberate monosaccharides. That's quite important. And I would also say that the monosaccharides are themselves reducing sugars. All you then have to do, which is, please don't forget, is that because you've added acid, you need to neutralize the acid, usually with sodium hydroxide. You can use sodium hydrogen carbonate as well, and just add that slowly, spatula-wise. And then you repeat Benedict's again. If I do it in yellow, you've got a negative result to start with. Actually, you might not see that. Negative result to start with, but you then hydrolyze the glycosidic bond and you end up with a positive result at the end. So this then is a positive result that you can see, and that allows you to infer that you have a non-reducing sugar, as opposed to just doing Benedict's the first time and using the negative result as evidence. You cannot do that. It's not good science, because it could have just not worked for some other reason. Right, that's the complicated one done. So that's testing for reducing and non-reducing sugars. We're now just going to look at how to test for starch, which is even easier. And this whole test relies on the fact that we use iodine solution. And iodine solution is made up of iodide ions, I-. And specifically, when you add this to starch, don't forget starch is made up of amylose and amylopectin. Hopefully what you remember is amylose is a linear molecule. It coils up because of intramolecular hydrogen bonds. Intra meaning within a molecule. Intramolecular hydrogen bonds, as opposed to something like cellulose, where we've got intermolecular hydrogen bonds between the linear molecules. Don't forget, hydrogen bonds would be sort of between bits like this. Don't read too much about that. Amylopectin, on the other hand, I'll do it in red, is branched, very much like glycogen. So it's kind of linear to a point. We've got an alpha-1,4 glycosidic bonds, but we also then can have 1,6 glycosidic bonds, which allow a linkage group to come off here. You might have one here, you might have one here, etc. And we're more focused on amylose actually for this. And because of its coiled structure, iodine, I'm going to do the color slightly deliberately. Iodine is a yellow, or iodine solution is yellow. Okay, so here we go. We've got iodine solution. Here. And when we add this to our amylose, the iodide ions actually interact in threes. So we have I- 3 times. And this forms a complex of I3- ions. That's kind of what we call it. Oops, that's horrible. Essentially, these can intercalate within or between coils. So you might get 3 of them here, 3 of them here, 3 of them here, 3 of them here, right? Zooming into each one of these dark blue blobs, of course, we have 3 individual iodide ions, as you can see here. And what that does is it changes the color because it alters the sort of chemical properties and the wavelength of light that is absorbed by this complex. And it means that the iodine solution will turn from yellow to blue-black. That's what we say. It becomes a blue-black solution. And that's all you need to know. And that's shown here. So actually, it's shown here is your triiodide group here. So you've got 3 iodide ions, and it is interacting within the coil, within the coil of the glucose molecule, the alpha glucose molecules in amylose specifically. And you can see the coil kind of wraps around the triiodide ion, altering the sort of light scattering properties of this molecule and the wavelength of light that is absorbed by it. So this is shown here, where if you're going to do this on a leaf, and this is one just to bear in mind, you need to decolorize the leaf, because if you're going to add dye to it or iodine, you don't want any pigments in the leaf causing aberrant results. So the first thing you actually do for a leaf is you boil it in water. You then add ethanol to decolorize it, and you then leave it in cold water, and you place the decolorized leaf on a Petri plate, and you add a few drops of iodine solution, which is yellowy orange. If you don't have starch, then it remains yellowy orange. If you do have starch, as we said, the iodine solution turns blue-black, meaning that starch is present. So if you're going to put this in an exam, you only literally need to say a few points, which is you add a few drops of iodine solution to the sample. It can be a solid sample as well for this one. A positive result means the iodine solution goes from yellow, which you do have to write, to blue-black. So we go from yellow solution to blue-black. And that is your positive result. That's it. That's your 2 tests. You need to know how to test for starch using iodine, how to test for a reducing sugar using Benedict's, that's the straightforward version, and how to test for a non-reducing sugar using Benedict's that gives you a negative result to start with. But don't forget, you cannot use the negative result. You have to then hydrolyze the glycosidic bond and then retest using Benedict's to then get a positive result. Cool. Thank you very much.